Welcome everyone. Today's presentation is about Staph aureus. This is a very notorious, very common pathogen that we encounter daily in our microbiology labs. And after going through this presentation, you will be able to enlist the common infections caused by this organism, describe the general characteristics of this organism. You will be able to identify as well as perform the antimicrobial susceptibility testing in the lab. So let's start with our first slide. So when we talk about Staphylococcus aureus, what they are gram-positive cocci, and as the name indicates, the staphyle, which means that they are arranged in clusters, like the clusters of grapes. They are all catalase positive. If we talk specifically about aureus, it is coagulase positive, which differentiates it from other staphylococci. So the coagulase negative staphylococci form a larger group of staph, but they are clinically less relevant, and Staph aureus is the main pathogen in this genera. These are non-motile and non-spore forming cocci, and they are heat tolerant as well as they have the ability to grow in the presence of high salt concentrations. So a little about their pathogenicity. As high as 30% of healthy population carries Staph aureus. Most of the colonization is on the skin, primarily in the anterior nares where they are localized. And if we do the screening for MRSA or Staph aureus as a general screening in the population, we will find that almost one third of the population is carrying Staph aureus. Moving on, it can cause the skin and soft tissue infections. If the infection goes deep, it can have serious consequences. And hospital acquired infections are always causing the increased morbidity and mortality if the causative organism is Staph aureus. For surgical site infections, an infection will lead to the lengthy hospital stay, which will lead to the increased hospital costs. Moving on to the lab identification, the primary appearance of Staphylococcus aureus on sheep blood agar is the classic beta hemolytic colonies after 24 hours of incubation. And the aureus, which means the gold color, is not typically seen in all the strains. Some wild type strains we do see in our clinical lab sometimes the classical picture of Staph aureus, but most of the isolates are like the one presented in this picture. They don't carry a aureus color, but they do carry a double zone of hemolysis mostly, and the hemolysis is more pronounced in the carbon dioxide. And however, be careful because some of the strains are non-hemolytic. So simply based on the colony morphology, we should not decide aureus or coagulase negative staph. We should do proper testing before declaring an organism as staph aureus or coagulase negative staph. Further on to the lab identification, if we do the gram stain, we will see gram positive cocci, which are arranged in clusters. And as we previously talked, the staph means the bunch of grapes, they are arranged in a cluster shape. The catalase enzyme produced by Staph aureus and other bacteria, it will hydrolyze the hydrogen peroxide and produce water and oxygen. And this oxygen will be visible as the bubbles coming out of the hydrogen peroxide solution, and it indicates a positive reaction. And the coagulase test is like a sort of preliminary confirmatory test that we can rely on and report that probable Staph aureus has been isolated. What happens is that the coagulase produced by Staph aureus, it converts fibrinogen to fibrin, and the coagulase is either bound to the cell wall of the Staph aureus or it is produced as a free form. So the bound coagulase can agglutinate plasma on a slide test, while if it's only free coagulase, it will not coagulate the plasma on the slide. So to detect the free coagulase, we need to go to the tube method, which detects the free coagulase. The only limitation is that the tube method requires at least 18 to 20 hours of incubation before declaring it negative. So it's not really used in the lab nowadays, this plasma based coagulase test. What we use nowadays is a latex test, which detects the protein A and the bound factor. The bound factor is the same as coagulase, but the protein A is an integral part of most of the Staph aureus. Almost 95% of the Staph aureus have protein A in their cell wall, and this protein A is capable to bound with the Fc portion of IgG. So what we have done is that we have coated the latex particles with both fibrinogen and IgG. So if the tested isolate will have a bound factor or protein A or both of them, they will quickly agglutinate all these latex particles, which will be very clearly visible. And this test has really revolutionized the coagulase testing, which was before tested on tube and slide method, and the results were not readily available for the tube method. Moving on to the identification, nowadays the commercial ID systems from Vitek, Microscan, Phoenix, and the latest Malditov, they are able to reliably identify Staph aureus without any doubts. For the susceptibility testing of Staph aureus, for all beta-lactam agents, we need to test only penicillin and oxacillin. And actually for oxacillin, we can use cefoxitin. So testing only penicillin and cefoxitin will give you the results for all beta-lactam agents. So if a Staph aureus strain is susceptible to penicillin, it's taken as susceptible to all beta-lactam antimicrobials. While a penicillin resistant strain is resistant to penicillinase labile penicillins, which are amoxicillin, ampicillin, ticarcillin, and piperacillin. Alright, moving on to the oxacillin. If we test oxacillin and we find that the Staph aureus is sensitive to oxacillin, we can reliably predict that this Staph aureus is going to be sensitive to cloxacillin, dicloxacillin, methicillin, nafcillin, and all beta-lactam combination agents, for example, amoxiclav, ampicillin sulbactam, piperacillin tazobactam, all the oral cephems, for example, cefaclor, and parenteral cephems, for example, ceftriaxone, cefotaxime, and carbapenems, doripenem, ertapenem, imipenem, and meropenem. So the methicillin susceptible and methicillin resistant Staph aureus are actually those Staph aureus which are either susceptible or resistant to oxacillin. So the Staph aureus which is resistant to oxacillin, they are termed methicillin resistant Staph aureus and are considered resistant to other beta-lactam agents, beta-lactam combination agents, and carbapenems. Since the testing for oxacillin requires the addition of 2% sodium chloride to the media, so most of the labs they prefer not to test oxacillin, but they use a surrogate marker, cefoxitin, which is equally reliable to detect the methicillin resistance in Staph aureus. And there are other ways to detect oxacillin resistance is to either directly detect the MEC-A gene of Staph aureus using the PCR technique or detect the product of the gene, that is penicillin binding protein 2 prime. So the MACA gene detection is quite common now in the labs. Most of the labs have a closed system PCR in the labs, and they detect and report this within few hours. So the results are readily available to clinicians for effective management of the patients. Okay, moving on with the susceptibility testing. The vancomycin susceptibility is not reliable using the Kirby-Ward distribution method. We should only use the e-test, broth dilution, or agar dilution method to test for vancomycin. And any isolate which is intermediate or resistant to vancomycin must be sent to a reference lab for confirmation. So this was all about the Staphylococcus aureus, the routine testing in the microbiology lab. And please don't hesitate if you have any questions, suggestion, any comment. Please write it to me, and I'll be glad to answer your questions. Do like and subscribe my channel. I'll be uploading more videos for microbiology. Have a nice day.